Researchers from Germany have developed the world's smallest ultrasound detector. It is based on miniaturized photonic circuits on top of a silicon chip. With a size 100 times smaller than an average human hair, the new detector can visualize features that are much smaller than previously possible, leading to what is known as super resolution imaging. Since the development of medical ultrasound imaging in the 1950s, the core detection technology of ultrasound waves has primarily focused on using piezoelectric detectors, which convert the pressure from ultrasound waves into electric voltage. The imaging resolution achieved with ultrasound depends on the size of the piezoelectric detector employed. Reducing this size leads to higher resolution and can offer smaller, densely packed 1 or 2 dimensional ultrasound arrays with improved ability to discriminate features in the imaged tissue or material. However, further reducing the size of piezoelectric detectors impairs their sensitivity dramatically, making them unusable for practical application. Silicon photonics technology is widely used to miniaturize optical components and densely pack them on the small surface of a silicon chip. While silicon does not exhibit any piezoelectricity, its ability to confine light in dimensions smaller than the optical wavelength has already been widely exploited for the development of miniaturized photonic circuits. The researchers capitalized on the advantages of those miniaturized photonic circuits and built the world's smallest ultrasound detector, the Silicon Waveguide Echelon Detector or SWED. Instead of recording voltage from piezoelectric crystals, SWED monitors changes in light intensity propagating through the miniaturized photonic circuits. This is the first time that a detector smaller than the size of a blood cell is used to detect ultrasound using the silicon photonics technology. If a piezoelectric detector was miniaturized to the scale of SWED, it would be 100 million times less sensitive. The SWED size is about half a micron. The size corresponds to an area that is at least 10,000 times smaller than the smallest piezoelectric detectors employed in clinical imaging applications. The SWED is also up to 200 times smaller than the ultrasound wavelength employed, which means that it can be used to visualize features that are smaller than 1 micrometer, leading to what is called super resolution imaging. As the technology capitalizes on the robustness and easy manufacturability of the silicon platform, large numbers of detectors can be produced at a small fraction of the cost of piezoelectric detectors, making mass production feasible. This is important for developing a number of different detection applications based on ultrasound waves. While the researchers are primarily aiming for applications in clinical diagnostics and basic biomedical research, industrial applications may also benefit from the new technology. The increased imaging resolution may lead to studying ultrafine details in tissues and materials. A first line of investigation involves super resolution optoacoustic imaging of cells and microvasculature in tissues. But the SWED could be also used to study fundamental properties of ultrasonic waves and their interactions with matter on a scale that was not possible before.